life cycle of the angiosperms दुईटा phases चाहिँ हुन्छ त्यसको एउटा नाम चाहिँ हो sporophyte sporophytic plant body त्यो plant जुन चाहिँ हामीले देख्छ which we can touch see त्यो त्यसको root stem leaf सबै यिनीहरु चाहिँ के हो that is the sporophyte and all the cells of the sporophytic plant body मा चाहिँ diploid cells हुन्छ they have two sets of chromosomes so they are sporophytes अब sporophyte को काम चाहिँ के हो sporophytic plant body ले चाहिँ के गर्छ they produce spores sporophyte means it is producing the spores okay so spores जुन चाहिँ छ तर the spores produced are haploid I told you sporophyte is a diploid organism diploid plant body but your spores produced are haploid so यहाँ कुन चाहिँ division हुनु पर्छ भन्दा meiotic division ले चाहिँ spores produce गर्छ which are haploid now this spores will divide and redivide to form another type of plant body that is known as your gametophyte okay so this gametophyte plant body cha त्यसको plant body it is developed from the haploid spores so therefore the plant body will also be haploid okay so gametophyte plant body ko kaam चाहिँ के हो to produce the two different types of the gametes male gamete र female gamete produce गर्नु okay so when the two male and the female gametes fertilizes to form the diploid first cell that is the zygote this zygote will again यो diploid cell भयो okay so यो diploid cell चाहिँ के हुन्छ redivide mitotically to form the entire sporophyte okay so this is the general life cycle of the angiosperm so this is also known as the alternation of generations which you've studied in your junior classes the sporophyte gives rise to the spores germinate to form your gametophyte gives rise to the gametes fuse to form the sporophyte okay so this is the life cycle now angiosperms are heterosporous that means angiosperms ले चाहिँ दुईटा type को spores produce गर्छ एउटा spores लाई two types of spores are produced one type of spore is known as your microspore एउटालाई भन्छ microspore अर्कोलाई भन्छ megaspore two types of spores are produced by the sporophytic plant body and the कुन चाहिँ division पर्छ meiosis gives rise to the two different types of spores now microspores ले के गर्नुपर्छ it will give rise to a gametophyte so this microspore बाट निस्केको gametophyte लाई चाहिँ we call it the microgametophyte we call it the microgametophyte okay and the megaspore also gives rise to another gametophyte त्यो gametophyte को नाम चाहिँ के हुनु पर्छ त्यो gametophyte को नाम हो megagametophyte so megagametophyte is also known as the female gametophyte microgametophyte is also known as the male gametophyte in flowering plants your microgametophyte चाहिँ के हो microgametophyte is the pollen grains okay so this is very very important it is the pollen grains that is formed त्यो चाहिँ microgametophyte हो okay megagametophyte चाहिँ के हो ovary भित्र ovule थियो ovule भित्र there is a female gametophyte formed त्यो gametophyte को नाम चाहिँ हो embryo sac the female gametophyte is known as the embryo sac this is important name of female gametophyte and male gametophyte say pollen grain okay so the male pollen grains will produce what pollen grains ma ke produce bhako huncha the male gamete is produced what is produced by the megagametophyte embryo sac produces the egg okay so they fuse together to form the zygote now the zygote develops into the sporophytic plant which is diploid okay so this is a general life cycle of your flowering plants now if we are to see the development of the male gametophyte or the pollen grain चाहिँ कसरी develop हुन्छ भन्दाखेरि in the flower यो male gametophyte कहाँ develop हुन्छ it develops in the stamens now if you take one stamen okay and which part of the stamen मा हामीले focus गर्नुपर्छ भन्दा it is the anther the top portion the head portion, not the filament. यो anther लाई चाहिँ हामीले काट्यो, त्यसको section काटेर हेर्यो भने चाहिँ you can see this type of structure. Now the anther is a two-lobed structure. Okay, so it is a two-lobed structure. So यो lobes हालाई यो anther लाई यसरी TS काट्दाखेरि हामीले we can see 4 compartments. Okay, so there are 4 compartments like this. So this is your anther wall. यो सबै यो inner चाहिँ यो चाहिँ anther wall भइगयो. Okay, so this is the anther wall. There are 4 compartments here. Okay, त्यसलाई चाहिँ के भन्छ? यो 4 compartments लाई चाहिँ we call it the microsporangium. We call it microsporangium. Okay, or later we can also call it the pollen sac. जहाँ चाहिँ pollen formation हुन्छ. So it is first known as the microsporangium. Okay, and the wall of the anther, yellow portion जुन चाहिँ छ, the wall of the anther को चाहिँ innermost wall लाई चाहिँ we call it the tapetum. Okay, so the wall that surrounds this sporogenous tissue त्यसलाई चाहिँ भन्छ tapetum. So यो tapetum is important because यो जब microspores develop हुन्छ त्यसलाई nutrition दिनुमा help गर्ने tissue को नाम चाहिँ हो tapetum. This yellow portion is known as your tapetum. This portion. Okay, and this this tissue that is present inside enclosed by the tapetum, यसलाई चाहिँ हामी भन्छ sporogenous tissue. Okay, from where the spores will be produced. Okay, so this is the sporogenous tissue that is present. So we take this one sporogenous tissue, okay, and यसको cells जुन चाहिँ छ यो sporogenous cells यो tissue को यो cells लाई चाहिँ we call it if you take one cell, this is known as the microspore mother cell. Why is this known as the microspore mother cell? Because it gives rise to the microspore. So this microspore mother cell is diploid. So your sporogenous tissue ma jatti pani cells haru cha sabaima they have 2 sets of chromosomes, so they are diploid. Okay, so this is the microspore mother cell. Now your microspore mother cell ma चाहिँ के हुनु पर्छ? It has to undergo a meiosis. A meiotic division हुँदाखेरि चाहिँ you will get a spores, 4 spores. Now you know during the meiosis, during meiotic divisions, you will see one diploid cell will give rise to, during the meiosis, one diploid cell will give rise to, during the first meiosis हुँदाखेरि it gives rise to 2 cells, no? So those 2 cells are but के भएको हुन्छ त्यसको ploidy level चाहिँ reduce भएको हुन्छ. That means your 2 cells formed during after the first meiotic division, this will be haploid. Okay, then after that meiosis फेरि second meiotic division हुँदाखेरि चाहिँ each of these haploid cells will divide, okay, will make copies of itself. So by the end of meiosis, what will you find? By the end of meiosis you will find 4 haploid daughter cells are produced from one diploid cell. So this is very important. You will get 4 cells, but the 4 cells त्यसको ploidy level चाहिँ घटेको हुन्छ. त्यसको यो diploid cell हो भने यसको chromosome set चाहिँ घटेको हुन्छ. So there are 4 daughter cells. So here meiosis your mother cell जब divide हुन्छ, so you will find by meiosis your 4 cells are produced. Okay, so the 4 microspores that are produced, that is formed here, यसलाई चाहिँ हामी microspore भन्छ. Okay, so the 4 microspores are all haploid. यो चाहिँ haploid हो. Microspore mother cell चाहिँ diploid थियो. अब यसको ploidy level चाहिँ घट्छ after meiosis. So this is now known as the microspore. तर यो microsporeहरु यहाँ जस्तै separate cells भएको छ. So one cell gives rise to 4 separate cells. So यसमा चाहिँ तर first time मा पहिल्यै सुरु चाहिँ microspore form हुँदाखेरि separate microspores form हुँदैन. They are all clumped together, grouped together. So this 4 cells grouped together त्यसलाई चाहिँ हामी भन्छ microspore tetrad. We call it tetra, means 4. So it is known as the microspore tetrad. Okay, so later what happens is this microspore will also start to the microspores tetrads ko jun chai microspores cha individual microspores tyo pani separate huncha. Now see, all of them are haploid. Okay, so after the microspores are formed or separated, then certain changes occur in the microspore. First चाहिँ के आउँछ भन्दाखेरि there is one development of a wall. Two walls are formed around the microspore. Okay, so the inner wall that is formed here, this white portion here, this is known as your intine. Inner wall, intine. This is made up of cellulose. Whereas the outer portion that is formed, this is known as your exine. Outer layer formed is known as the exine. Now this exine is very, very important because this exine is made up of a very, very important organic resistant material. This is known as the sporopollenin. Now this is very important. What is sporopollenin? Sporopollenin भनेको चाहिँ एउटा highly resistant organic material हो, chemical हो. This यो जुन चाहिँ exine ले बनेको, exine चाहिँ sporopollenin ले बनेको हुन्छ. So यो exine or यो sporopollenin चाहिँ के हो भन्दाखेरि it is very, very highly resistant to all types of your degradation. Temperature ले पनि त्यसलाई degrade गर्न सक्दैन. It's very, very high and tough. So temperature ले गर्न सक्दैन, acids ले गर्न सक्दैन हो, त्यसमा कुनै टाइप को microbial attacks हुनु सक्दैन. So त्यसले गर्दाखेरि चाहिँ यो pollens earlier ancient plants हरुको pollens हरु चाहिँ well preserved भएको कारण चाहिँ के हो, because there is the presence of a very highly resistant chemical which is known as the sporopollenin. So त्यसले गर्दाखेरि this sporopollenin is very, very important. You must remember it is not easily degradable. So then after the formation of the sporopollenin, now your sporopollenin ma ni एउटा certain portion एउटा त्यो sporopollenin चाहिँ सबैपट्टि uniform हुँदैन. At certain points the sporopollenin is not found. So this spore that is left here, त्यसलाई चाहिँ हामी भन्छ germ pore. Okay, त्यहाँबाट चाहिँ यो किनभने यो male gametes form भएपछि यो male gametes चाहिँ यो there is a formation of a tube like structure. त्यसलाई चाहिँ हामी pollen tube भन्छ. Later on we'll study. So त्यति बेला चाहिँ यो germ pore बाट निस्किन्छ यो pollen tubes. So त्यसले गर्दाखेरि sporopollenin is very, very tough. त्यसलाई degrade गर्न सक्दैन, break गर्न सक्दैन. So त्यसले गर्दाखेरि एउटा certain region मा चाहिँ त्यो sporopollenin ले भएको हुँदैन. So त्यसलाई चाहिँ हामी भन्छ germ pore. Okay, so if you can see the exine out here smooth छैन. Intine is smooth, but exine out here is very, very, you can see it is very rough. It has many ornamentations. हेर्दा पनि यो pollens हरु हेर्दा पनि you can see of different sizes. Different species of plants will have different sizes and shapes, colors, and also त्यसको बाहिरको pattern, exine को pattern हरु पनि different हुन्छ. So after the formation of this microspore, okay, this pollen अब यो formation भएपछि, now what happens is the cell, this microspore मा भएको जुन चाहिँ nucleus छ, त्यो nucleus divides mitotically. This nucleus divides. Now this nucleus is haploid. Remember, this nucleus is haploid. So haploid भनेको माने mitosis मा के हुन्छ? So J haploid level, ploidy level change हुँदैन. So if you have 2N mitosis भयो भने यो mother cell बाट you will find 2 daughter cells, and both of the 2 daughter cells are only, they have the same ploidy level, no change. If this is N, the mother cell is N, mitosis भयो भने you will find 2 daughter cells, त्यसको तर ploidy level change हुँदैन. So it is N only. So here also यसमा पनि N छ. So you will find another nucleus, haploid nucleus that is formed. Okay, so haploid nucleus जब form भएपछि Okay, so there is a formation of a haploid nucleus. Now after the formation of the haploid nucleus, there is a cytokinesis. Nucleus division keratokinases भयो. अब cytokinesis हुँदाखेरि चाहिँ unequal type of cytokinesis हुन्छ. That means त्यो दुईटा cells जुन चाहिँ form हुन्छ after the mitotic division equal, यस्तो equal cells, equal size को cells हुँदैन. It is of two unequal sized cells. So cytoplasm is laid down here. Okay, so you will find two unequal sized cells. So now we'll name the cells. The larger cell is known as the vegetative or the tube cell. The smaller cell is known as the generative cell. Okay, so this vegetative cell or the tube cell is very important because you know that time I already told you pollen tube भन्ने निस्किन्छ भने मैले during pollen germination pollen tube निस्किन्छ germ pore बाट भनेको छु. So त्यो germ pore मा यो pollen tube माने produce हुनुको लागि चाहिँ which cell is important भन्दाखेरि this larger cell, that is vegetative cell or the tube cell, that is important for formation of the pollen tube. Okay, so pollen tube ko lagi tube cell or the vegetative cell, remember that. Next is your generative cell. Now your cell, this small generative cell le chai ke garcha, it will further later on give rise to your two male gametes, that is the two sperms are produced here. Okay, so now what we can see here is जब पोलन्स हामीले एन्थर बाट बर्स्ट भएर निस्किन्छ, जब पोलन्स बर्स्ट भएर निस्किन्छ त्यो बर्स्ट भएर पोलिनेसन भन्छ जुन चाहिँ प्रोसेसलाई, त्यो पोलिनेसन प्रोसेसमा चाहिँ जब पोलन्स हैव टु रीच दी हैव टु लीव द एन्थर्स ओके एन्ड गो टु द स्टिग्मा, त्यति बेला द पोलिनेसन प्रोसेस हुनु अगाडि चाहिँ यो के अरे टु सेल स्टेजमा हुन्छ. Pollens will be at the two cell stage मा चाहिँ pollination हुन्छ. So and after that यो जुन चाहिँ some majority of the angiosperms मा चाहिँ two cell stage यो pollen grain or the male gametophyte two cell stage मा हुँदाखेरि चाहिँ त्यति बेला चाहिँ pollination हुन्छ. Okay, so and some angiosperms मा चाहिँ फेरि यो pollen के गर्छ, before the pollination starts only यो pollen के गर्छ, यो pollen grains के हुन्छ, यो generative cell will divide into two male gametes. Now you have a three cell stage. So यो three cell stage मा some angiosperms ले चाहिँ three cell stage मा pollination गर्छ, sometimes चाहिँ two cell stage मा चाहिँ pollination हुन्छ. So that is your the overall development of the male gametophyte. The male gametophyte produced here is the pollen grains. Okay, so the pollen grains are produced where? Inside the anther. Which part of the anther? Vandakiri. Inside these pollen sacs, यहाँ चाहिँ anthers produce हुन्छ. Okay, so once it matures, your mature pollen grain भएपछि चाहिँ the anthers will split open or dehiscence हुन्छ and the anthers will be released. Okay, so they will be released either in the two cell stage or the generative cell may again divide once mitotically to form two daughter cells. Okay, male gametes, and then in the three cell stage the pollination may also occur. Right, now we will start with the formation of the female gametophyte. Now the female gametophyte in the flowering plants, embryo sac, we call it embryo sac. Now where is this embryo sac formed? Embryo sac is formed inside the swollen structure. This swollen structure the carpel ko swollen basal end we call it the ovary. Your ovary bhitro there is a structure which is known as the sporangium. Your sporangium ko naam ke ho? Ovule. Your ovule bhitra chai there is the formation of the female gametophyte. Okay, so now first before we go on to the formation of the gametophyte, let's see the structure of this ovule. Your ovule, this megasporangium चाहिँ कस्तो हुन्छ? जहाँ चाहिँ त्यो स्पोर फर्मेसन हुन्छ त्यो मेगास्पोरेन्जियम चाहिँ कस्तो हुन्छ भन्दाखेरि you find that this is the ovule. Now ovule is surrounded by two layers, two protective coverings. यो दुईटा protective covering लाई चाहिँ हामी we call it integuments. Okay, the outer and the inner integuments. Now inside the integuments, enclosed inside the integuments is the general tissue. The general tissue of the ovule is known as the nucellus. We call it the nucellus. Okay, so this ovule is attached to the swollen portion. Okay, it is attached to the swollen portion by a stalk. This stalk, this is known as the funicle. Okay, so this funicle attaches this ovule with the placenta of the ovary. Alright, so that will not concentrate on. So next what we have to learn here is your integuments say यो nucellusलाई cover सबैपट्टि गर्छ तर at a certain region it is not enclosing the nucellus. So this portion where the integument does not enclose the nucellus fully, this portion is known as the pore is known as the micropyle. This is very important, micropyle, because it is through the micropyle that the pollen tube enters. Okay, so the micropyle is the pore, okay, that is formed by the incomplete enclosure of the integuments. Now the next end, the opposite end of the micropyle, this basal end, okay, the broad basal end of the ovule, यसलाई चाहिँ हामी chalaza भन्छ. We call it the chalazal end. Okay, so this is the main structure of the ovule. Now where is the formation of the embryo sac or the female gametophyte in the ovule भन्दा चाहिँ यो nucellus region मा, in the nucellar region, any cell of the nucellar region, okay, so this cell, any cell of the nucellar region, यो nucellar region को cell लाई चाहिँ any cell that functions as the it functions as the megaspore mother cell. Why? Because this mother cell will divide to form the megaspores. Okay, so this nucellar cell, any cell of the nucellus can act as the megaspore mother cell, okay, and divide to form the megaspores. Okay, so again we already told you that the sporophyte ma chai there is a meiotic division. Meiotic division le chai spores dincha bhaneko cha हामीले. So in this case we have two types of spores, megaspores and microspores. How is the megaspores produced bhanda? This megaspore mother cell, which is the tissue of this nucellus, will divide meiotically by meiosis. It will divide, okay, and form 4 haploid cells. This megaspore mother cell was diploid. It had 2 sets of chromosomes. Now by meiosis it will form 4 cells and all the cells are haploid. Okay, so out of the 4, these are the megaspores. Now we call it the megaspores. Now you megaspores ma chai the spores have to again germinate or produce the gametophyte. अब यो gametophyte produce गर्नुको लागि चाहिँ के हुन्छ? यो megaspores चाहिँ first चाहिँ के हुन्छ? Out of the 4 megaspores, only 1 megaspore stays, whereas the rest 3 degenerates. So this degenerates. Okay, so only 1 megaspore can function, okay, as the functional megaspore. And remember, this is haploid. Now you have a haploid megaspore, functional megaspore. Now what happens after that? Now there are a number of mitotic divisions occur. Mitotic karyokinesis. अब karyokinesis हुन्छ, nuclear division पुरा हुन्छ, free nuclear division हुन्छ. That means free nuclear division means the nucleus divides into many nuclei but does not divide, but cytoplasm does not divide. Okay, so this nucleus, haploid nuclei ma ke huncha? Mitosis huncha. So from 1 haploid nuclei you will get 2 haploid nuclei by mitosis. Okay, फेरि उयो हुँदैन, karyokinesis मात्रै छ, cytokinesis हुँदैन. Again the 2 nuclei यो दुईटा न्यूक्लियाई जुन चाहिँ फर्म भएको छ सेन्टरमा यो this will move towards the opposite poles. It moves towards the opposite poles. Then it starts to again divide mitotically. Feri from 2 cells you will get 4 nuclei are formed. All of them are haploid. 4 haploid nuclei are formed. From 4 haploid nuclei again mitosis huncha. So there are 3 sets of mitosis. 3 palta mitosis hunदाखेरि you will get the megaspore here has how many nuclei? It has 8 nuclei by mitosis. Okay, so remember till now there is no division of the cytoplasm. Okay, so you have 8 nuclei. Now what happens? 4 on one side, 4 on the other side. All are haploid. Now what happens? 2 of them, 2 from either ends, त्यो दुईटा न्यूक्लियाई चाहिँ बीचमा हुन्छ. यो दुईटा न्यूक्लियाई बीचमा हुन्छ. Alright, so you will find 3 nuclei on one side, 3 nuclei on the other side, बीचमा चाहिँ 2 nuclei. Right? After this, now the cytokinesis starts. Okay, so you will find the 3 cells are formed. Okay, on one side, then another 3 cells are formed on the other side. Okay, so the 3 cells on this side चाहिँ this end of the gametophyte चाहिँ कहाँ form हुँदैछ भन्दा this end is on the chalazal end. Chalazal end. This side यो पट्टि gametophyte चाहिँ के form हुन्छ भन्दाखेरि त्यसलाई antipodal cells भन्छ. Okay, the cells that are formed on the chalazal this side end त्यसलाई चाहिँ भन्छ antipodal cells. The cells that are formed on the opposite micropylar end, यो end मा form हुने cells लाई चाहिँ हामी भन्छ, we call it the egg apparatus. It is known as the egg apparatus, and out of the 3 cells in the egg apparatus, दुईटा लाई चाहिँ यो दुईटा surrounding cells लाई चाहिँ we call it the synergids. It's known as the synergids. बिचको cell लाई चाहिँ we call it the egg cell. Okay, so this is the egg cell. So egg cell and the synergids, they compose the egg apparatus. Now what about the 2 nuclei that is present here? So bichma jun चाहिँ यो nuclei छ, यो 2 nuclei, यो 2 nuclei लाई चाहिँ we call it the polar nuclei. We call it the polar nuclei, and sometimes later on what happens, यो दुईटा polar nuclei can fuse together to form one. So N and N fuses together to form a diploid nucleus. त्यसलाई चाहिँ we call it the secondary nucleus. So if the polar nuclei, दुईटा polar nuclei fuse भयो अरे, N and N fuses together, it forms a diploid nucleus. The 2 nucleus लाई चाहिँ हामी भन्छ secondary nucleus. Now secondary nucleus fertilization को लागि secondary nucleus याद गर्नु एकदम important छ, किनभने यो secondary nucleus बाट नै endosperm is produced. Okay, it helps to form the endosperm, which is the nutritive tissue. Next one is the egg. Your egg cell which is present in the egg apparatus. This is also very, very important because the male gametes fuses with the egg during the fertilization process. So here what did we see? The spores, the megaspore, functional megaspore, it divided to form your gametophyte. Okay, so it formed your gametophyte. Now the gametophyte like produce gareksa, egg produce gareksa. In case of your micro gametophyte or the pollen grain, tesma ke produce bhako thyo, vegetative nucleus and 2 male gametes produce bhako thyo. Yes, so here the female gametophyte or the embryo sac. This is known as the embryo sac. This total structure is known as the embryo sac. Your embryo sac ma chai ke produce bhako cha, it produces the egg. Okay, and remember the cells this side of the, it's important to note the side where the antipodal cells र यो synergid cells egg apparatus कहाँ form हुन्छ भन्दाखेरि chalazal end towards this side चाहिँ antipodal cells produce भएको हो, towards this side micropolar end मा चाहिँ के प्रोड्युस भएको एग एपारेटस प्रोड्युस भएको सो द embryo sac is produced here okay so this is your development of the female gametophyte or the embryo sac